feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Lulin School of Medicine Healthy Longevity webinar. It's a great pleasure to have you all with us today. Dr. Vijay Yadav is my guest. We will talk about how nutrients, in particular taurine, influences the aging process. Please use the Q&A function for all your questions, and I think his talk might trigger lots of them. As always, we are starting with a short presentation, this time by Jandria So. She is our PhD student at the Center for Health and Longevity, and she will talk about another supplement, and that is alpha-ketoglutarate. And she will talk how AKG might improve health in humans.
Thank you for the kind introduction. As mentioned, my name is Jandira. Today, I will be presenting a review paper on alpha-ketoglutarate dietary supplementation to improve health in humans. This paper was published in the journal Trends in Endocrinology in 2021 by Gyanwali et al. From our Center for Healthy Longevity. The aim of this review paper is to discuss the regulatory role of AKG in aging, as well as its potential therapeutic use in humans to treat age-related diseases. So to begin, AKG is a keto acid generated from glutamate deamination in the body and is a key molecule for cellular energy and protein synthesis. Today, AKG can be synthesized exogenously and produced in several forms. AKG functions as an antioxidant in nitrogen and ammonia balance, as well as in epigenetic and immune regulation. The first indication that AKG supplementation leads to enhanced lifespan was reported in C. elegans with up to 50% longer. The mechanism for lifespan extension was linked to the downregulation of mTOR kinase activity. A recent study has also documented the anti-aging effects of AKG in Drosophila that also mediates its effects by mTOR inhibition. 2% of calcium AKG extended lifespan and health span in both male and female mice by 9.6 to 12.8% and 16.6 to 19.7% respectively. In those mice, a decrease in systemic inflammatory cytokines was observed, suggesting AKG can suppress chronic inflammation and improve in several health outcomes such as frailty, hair loss, and maintenance of body weight. In humans, dietary AKG may improve protein metabolism and synthesis in older adults and have shown to reduce biological age by 8 years as measured by DNA methylation. We performed a literature search and summarized the effects of AKG on aging and age-related diseases in humans. In cancer, AKG regulates signaling pathways linked to oncogenesis, making it one of the potential anti-cancer agents. In the skin, topical application of AKG can diminish skin wrinkle formation by increasing collagen production. In severe burn patients, ornithine AKG significantly shortened wound healing time. And in the kidney, AKG regulates acid-base balance in renal tubules, and calcium AKG has been shown to improve kidney function and nutrition in chronic renal failure patients. In the liver, AKG can maintain nitrogen and protein balance and may be useful in treating non-alcoholic fatty liver disease and reducing brain ammonia and glutamate level in hepatic encephalopathy patients. In the heart and blood vessels, AKG has been shown to enhance myocardial oxidative capacity, increase energy production, and attenuate ischemic injury during heart surgery. AKG has been shown to reduce plasma lower density lipoprotein and cholesterol concentration in humans, implicating the metabolite in downregulating hyperlipidemia. In the brain, high concentrations of ammonia and nitrogen lead to several neurological symptoms, such as impaired memory. AKG can prevent the brain from oxidative damage associated with neurological diseases by increasing neuronal concentrations of antioxidative enzymes and quenching reactive oxygen species. Ornithine AKG in dogs showed increased brain oxygen and glucose utilization while reducing metabolic disturbances caused by hypoxia. Similar results were obtained in patients with stroke. And in the bone, AKG showed a protective effect against osteoporosis and in postmenopausal women with osteopenia. Oral calcium AKG for 6 months increased bone mass density. In the skeletal muscle, AKG increased muscle strength and endurance, in part by preventing muscle protein breakdown. Clearly, AKG has pleiotropic functions, and in aging, these have implications for its use as a longevity-enhancing mimetic. And in the gastrointestine, AKG can help in epithelial restitution and protection against stress-induced epithelial damage. Emerging evidence has shown that AKG supplementation might affect intestinal innate immunity through influencing intestinal microbiota and inhibiting intestinal villi apoptosis, hence maintaining gut integrity. So to conclude, AKG has been used in the treatment of several diseases in model organisms as well as in humans, as we have seen earlier. Yet many actions and mechanisms of AKG are not fully understood. Exciting results from preclinical studies indicate that mTOR is involved in human chronic diseases where modulation of mTOR activity by AKG may play an important role in aging. Hence, we recommend more research focusing on AKG as an anti-aging supplement to improve health span in humans. And with that, I'd like to thank you for your kind attention.
Thank you so much, Janjira. Great. So I think we are all looking forward to our own results of the ABLE study, where we also give AKG to healthy individuals being 40 to 60 years old. If you want to enroll, still there are places available. Now it's my great honor to introduce you to our guest. Dr. Vijay Yadav is today with us. He is Assistant Professor of Genetics and Development at the Columbia University, and he obtained his PhD at the Indian Institute of Science in India in molecular physiology. And he worked with lots of model organisms like mice, rats, even buffalo cows and monkeys, I learned, to understand ovarian regulation in primates. And for that work and other work, he received lots of awards and other very nice kind things. His group investigates now how the molecular setup of organisms changes during the aging process, with the overarching aim, as so many, to develop novel anti-aging interventions. His group has discovered how micronutrients such as vitamin B12 and more recently taurine regulates the physiology of our body and how deficiencies affect the aging process. It's really an honor because I know you just had the beautiful publication about taurine, and we are really looking forward, Vijay, to hear your talk. The floor is yours.
Thank you, Andrea, for the kind introduction. I would just open my slides. Give me a moment. So I'll start my slides with this image that says human society is really aging. Aging-related changes manifest as hallmarks of aging and lead to increased dysfunction of different organs and different diseases. Now, in my lab, we are interested in identifying how aging is leading to changes in molecular composition of organisms, because with age, you have a variety of changes, different hormones change, different metabolites change, different cellular processes change. So we aim to identify these molecules in order to identify interventions that can lead to better healthy lifespan. In our quest to identify such metabolites and molecules, we performed way back in 2012 a metabolomic screen in aged humans in a specific nutrient deficiency. And this analysis showed that variety of molecules were changing with age. One of the most dramatically down-regulated molecule in this screen was taurine. And this made us interested in taurine, what it is and what does it do. In fact, when my senior scientist in my lab, when she came up to me and said, do you know what taurine is? 13 years ago, I didn't know. That was my answer. And I told her, let us find out what does it do. So taurine is not a new molecule. It was identified way back in 1827, almost 200 years ago. For almost 140 years, it remained in hibernation because biochemistry and molecular biology was still in a very old age. It was still evolving. And in 1950s, what caught people's attention was when people became rich after World War II, and they needed some kind of a pet diet to be given to their pets in the morning when they went to office, so that when they come back from office, their pets are happy. This introduction of pet diets in the society led to increased incidence of diseases. So these were pellets which were produced by pet industry, given in big bags, and then you just drop some pellets in the bowl of the pet. Many diseases precipitated, such as cardiovascular abnormalities and growth defects in cats and dogs. One of the most striking aspects of this entire episode was that cats started turning blind. So they were visibly blind. They were hitting walls at home. They were dropping vases. So it was very distressing sight for the pet owners to see. They were rich households. Now they had a cat that is blind. This increased the interest in society to identify why the cat is turning blind. So it is kind of a cat story. Now, there was increased investment in research and development in science to find out why cats are visibly blind. This intense investigation almost lasted for 10 to 15 years. And in 1975, there was a seminal paper published in Science that showed that retinal degeneration or blindness in cats is caused by a one molecule deficiency in their diet, and that molecule was taurine. After that, cat food was supplemented with taurine and blindness went away overall. So this first episode of the blindness in cats brought taurine into limelight. Another 13 years, there was another study published in Science that showed that cardiac abnormalities in cats is also caused by taurine deficiency in their diets. These two studies, along with several others, hundreds of studies, led people to investigate more thoroughly what taurine is and what does it do. And now we know that taurine is associated with variety of health parameters in humans and other species. But what is not known when we were starting is that, does taurine affect the aging process? And that is the question we started to investigate. To investigate this question, we first measured taurine levels in variety of species. In mice, taurine levels declined with age. In monkeys, taurine levels declined with age. In humans, taurine levels declined with age. So these three species, mammalian species, told us that taurine levels are declining with age. Now, to find out whether this decline in taurine levels that happens with age is a driver of aging and actively participates in the aging process, or is it just a passenger of aging and is just going along for a ride, we then performed an intervention study in mice when the taurine levels were low. And we started this study at around 14 months of age in mice, which is equivalent to around 45 to 50-year-old humans. And after optimization of doses and frequencies, we gave these mice once daily taurine supplementation for life. This taurine supplementation of once daily led to increased median lifespan of females by about 12%. So the females lived longer. In males, this taurine supplementation increased lifespan by around 10%. So these males and females both were living longer. Now, this raised the question that mouse is just one model organism. How widespread this effect is. So to investigate that, at that point, I contacted Brian right at NUS, and then we started investigating in different species to try to see what happens in other species. And this collaborative network then identified that taurine supplementation in worms, that is another model organism, C. elegans, leads to around 23 to 30% increase in lifespan at a higher doses. In yeast, however, there was no effect. In a single cellular eukaryote, there was no effect of taurine supplementation on the replicative lifespan of yeast at a very high doses. Now, later on, we came to the realization that taurine metabolism and biosynthesis was still evolving at a very nascent stage in yeast in a single cellular eukaryote. And as the organisms evolved to multicellulars, to vertebrates, to mammals, this mechanism got more specialized and was imparting health benefits to different organisms. Now, the second question what we address is that, are these taurine-fed animals healthy? Because you don't want to be living longer and having a lot of diseases. To investigate that, then we next performed another experiment where we now supplemented mice with taurine. And after 1 year of daily supplementation, we measured variety of health parameters, measuring different organ functions. And this analysis showed that no matter where we looked at muscle, and the muscle strength was increased, neuromuscular coordination was increased, bone density was increased, bone functions were enhanced, pancreas functions by looking at glucose tolerance and insulin tolerance was increased, their GI motility was increased, and their brain functions were enhanced. They had a reduced anxiety and better memory. When we looked at immune system, now this aged immune system in taurine supplemented animal was behaving like a younger immune system, able to mount a better immune response. They had lesser adiposity in the body, and their energy expenditure was increased dramatically. Now, overall, these organ function analysis told us that the health span was increased in these taurine supplemented animals when these mice are equivalent to approximately 60 to 65-year-old humans. Now, conversely, then we next address that what will happen in an induced taurine deficiency. So taurine enters the cells through a transporter. One of the major transporter is SLC6A6. And we next knocked down this transporter in mice, and this led to decrease in taurine abundance in different tissues. And in liver, for instance, it was 50% decreased. In muscle, it was decreased by 90 to 95%. In the brain, it was decreased by approximately 70 to 75%. And across other organ systems as well. And this deficiency of taurine in a mouse model led to precipitation of variety of diseases in animals which were just 4 months old. And this increased morbidity of the animals led to their lifespan shortening by approximately 50%. So on average, mouse were living 32 to 34 months for life. And if you look at these taurine deficient, induced taurine deficient animals, they were now on, and the median lifespan decreased to approximately 18 months. So these studies of taurine gain of function by reversing the decline in taurine, which happens with age, and induced taurine deficiency in the mouse model told us that taurine deficiency curtails healthy lifespan. So in other words, taurine supplementation increases the median lifespan of mice and worms and increases health span of these rodents. Now, The next question was, how does taurine suppress aging phenotypes? To address this question, we used taurine deficient cells and performed a RNA sequencing analysis. And this RNA sequencing analysis told us that variety of gene signatures of aging hallmarks are clustered with taurine deficient transcriptome. Now, this was just a transcriptomic study. After that, we performed variety of different investigation in different species, in mice, in rodents, in zebrafish, in worms, and in monkeys, to see how these different hallmarks are interacting with each other. To me, it was not a surprise that different hallmarks are precipitating, because these hallmarks don't work in isolation, although we may tend to believe for different reasons, but these hallmarks all are coordinating this entire aging process in the cells. Now, to summarize this entire investigation, what we identified that taurine, once it enters the cell, it regulates variety of these features that get compromised with age. It was increasing stem cell numbers in few tissues. It increased autophagy in the cells, which goes down with age. It was improving proteostasis, and together it was improving nutrient sensing, the way cells were sensing different nutrients. It suppressed DNA damage. It also led to variety of changes in the epigenome, or DNA methylation and histone methylation. And in fact, when you look at the muscles of taurine deficient animals, their epigenetic age was increased by around threefold. So 60 to 80 weeks old animals were looking like to 210 weeks old animals. So, and it suppressed senescence dramatically, the accumulation of senescent cells or zombie cells that increase with age in our bodies. And it was suppressing inflammation. It was reducing the inflammatory markers in the blood, looking at different pro-inflammatory cytokines, for instance. So together, these studies told us that this nutrient, which is present in abundant amount in our bodies, is synthesized by liver primarily, and which can also be obtained in the diet, animal-based diet, or as a supplement, was affecting variety of features during aging and was improving the health of the cells or the vigor of the cells. Next, we looked at in humans. So in humans, we had access to around 12,000 subjects, longitudinal data for health. And we also had now the taurine and its metabolite levels measurements available. So we performed association analysis to see how does taurine levels in aged humans, these are around 60-year-old humans, associate with different health status. And this analysis showed that higher taurine levels in aged humans was associated with lower obesity. It was associated with lower BMI, lower waist to hip ratio, lower prevalence of type 2 diabetes. Their random glucose was lower. They had less liver damage. Their inflammatory markers in a high taurine containing individuals were lower, such as CRP. And variety of other parameters, such as hypertension, was lower. So these data were just associations, because in humans, it is not a causation. But this told us that a higher taurine abundance in the blood of aged humans is associated with better health of a human being. The next question was, okay, what about a healthy state of a human being? So what could be a better healthy state of a human being than exercise? And that is what we did next. We took different subjects, sedentary subjects, natural bodybuilders, marathon runners, and sprinters, to see if we put them through a bout of exercise on a bicycle, what happens to their taurine levels. And this analysis showed that no matter what individual we put on a bicycle, all these individuals had increased taurine levels after exercise. They had increased hypotaurine levels, a precursor of taurine, after exercise. They also had increase in the N-acetyl taurine levels after exercise. These data told us a bout of exercise increases taurine and its metabolite levels in the blood. Although we don't know the significance of this observation, to me, it seems to suggest that many of the health benefits of exercise could also be mediated by different molecules secreted in the bloodstream following exercise, such as taurine, which mediate the health benefits of exercise. Now, to come closer to humans, as close to possible, we performed a monkey intervention study next. And to do so, we took aged monkeys, which are equivalent to around 50-year-old humans, and perform a daily taurine supplementation in these monkeys. And after 6 months of daily taurine supplementation, we measured a variety of their health parameters. And this analysis showed us that the blue here is taurine supplemented monkeys, that they had reduced body weight gain, they had reduced adiposity, their bone density increased, they had reduced fasting glucose levels as a measure of glycemic index, they had less liver damage, and their immune system, the repertoire of immune cell, was behaving similar to a younger animal. And if one looks at a variety of oxidative damage markers in the blood of these rodent in monkeys, their DNA damage, oxidative DNA damage marker in the blood was reduced, the lipid damage marker was reduced, and their protein damage marker was reduced in the blood compared to control vehicle-fed monkeys. Now, together, these studies tell us that taurine intervention increases health in monkeys, which are closest species one can investigate to humans. Now, to summarize, what I have shown you today is that looking at yeast, worms, mice, zebrafish, monkeys, and humans, we could now collate all this information to tell that taurine deficiency during aging is a driver of aging. And I will end this talk with enormous number of investigators that helped us to investigate this important question, which took us around 12 years. And it started in my lab in Cambridge, and it ended up in New York City. But I really would like to thank especially Brian, who brought different organisms in picture, and it also tells us that how collaborative the aging field is, which is ready to help and ready to work together to address important questions, to identify interventions which can make us live longer and healthier. And the next step of this research is a clinical trial, which we are putting together across different countries and across different populations, because we do not know at the present time, first of all, what is the healthy reference range of taurine in different populations? How does it affect the aging process? We can measure different associations in different populations. And that is what, at the present time, we are invested into to start the clinical trial. Hopefully, by early next year, we should have all these funding and study design and populations in place to do so. And thank you very much for your attention. And I'm happy to take any questions.
Beautiful talk. Thank you so much. It seemed that taurine is the wonderful molecule who will all rescue us from the aging process in the end. But let me first ask you a question. What do you think is influencing taurine levels, and especially in humans? Can you elaborate a little bit more on it? Is it just the food we are taking influencing our taurine levels?
So it's a very important question in my view. We are intensively investigating this question. Now we have a few upstream regulators of taurine metabolism in aged humans. So with aging, we have this variety of nutrients which are changing. Not only these nutrients are changing, Our ability to absorb these nutrients is changing. Our ability to metabolize these nutrients are changing. Some of them are really essential nutrients, such as, for example, vitamin B12, which only comes from the animal-based diet. And vitamin B12 deficiency in aged human is very dominant. If you look at 60 to 65-year-old humans, the deficiency is very widespread. And it does not matter whether you're a vegetarian or a non-vegetarian. So it also tells us that the upstream regulators need to be identified. And I think we have some idea now for the upstream regulators, and hopefully there are a few studies which are right now in publication stage, and I cannot talk about it, but we have identified at least few upstream regulators which in humans lead to changes in taurine metabolism. And not only that, so if you look at just a diet now, so taurine can be obtained by diet, or it can be produced within our bodies. The ability of the liver, for example, the principal organ that produces taurine, also declines with age. And the third layer of complexity comes in picture, where the metabolism of taurine within the cell is being compromised. So if you look at the downstream taurine metabolites in aged humans in the blood or in animal models, we identified that there seems to be taurine resistance to the level of taurine present in the cells as well. So downstream taurine metabolites, such as, for example, mitochondrial tRNA metabolism, is severely compromised by 95% compared to in the liver, for example, taurine levels going down by just 60%. So there seems to be a variety of different hierarchy, from dietary changes to the absorption, to production of taurine in the body, to the way it acts in the cells, within the cells, to perform its functions.
Okay, let's disentangle that a little bit. So on the one hand side, we have diet, and on the other hand side, we have intrinsically produced taurine. If we just stick to the diet now, if I think remember correctly, it's especially coming from animal food. Do you already have any evidence that taurine levels, for example, are lower in vegetarians or vegans compared to meat eaters?
So it's a difficult question in my view to address, because a vegan diet also, variety of studies have been done that it is healthy. But we also know that that many of the molecules are not present in vegan diet. For example, B12. Vitamin B12 only comes from the animal-based diets because it cannot be produced in the body. So this study, along with others, tend me to think that nutritionists now need to look at diet more closely, because vegan people, for example, are given vitamin B12 supplement. Do we need to supplement with taurine? I do not know at the present time, but it is people who have had an intense vegan diet for 6 months to 1 year had 20 to 25% reduction in taurine levels. So maybe they have more resistance to taurine metabolism. So these questions, I think, will become more clearer as we go along. This took us around 12 years to do. And hopefully, we will have now, there is a lot of interest in taurine metabolism. And hopefully, in few years down the line, we'll have much more clearer picture of how taurine metabolism is changing in different populations. And as I said before, that we need to investigate, identify this healthy reference range in different populations before jumping on to the taurine intervention trial. And that is exactly what we are doing at the present time, to provide this healthy reference range across different ethnicities, across different countries, to figure out, okay, what is the appropriate level of taurine intervention that we will go in humans.
Yes, interesting. So you say, okay, don't jump if you are vegan now to eat products or meat products or take supplements, because we do not know that. That's thank you so much for that answer. But on the other hand side, you say, okay, we also intrinsically produce taurine in our liver. So you already, I think, showed us that taurine levels are associated with liver function or liver diseases or fatty liver we are seeing in obesity. Is that right?
Absolutely. Yes. So there are many animal studies also that have been done now in the literature that represent that fatty liver disease models, animal models, can be benefited by taurine supplementation. So there are indication that with the changes in liver function, you have changes in taurine ability of the body to produce taurine. And liver seems to be a very important organ to me. And one of the first evidence I always look at was in 1934 Nobel Prize that was given for feeding humans raw liver to cure pernicious anemia. And that we did not know what molecules were present in liver. And it took us almost 30 years later, we identified there was vitamin B12 in the liver that was curing pernicious anemia. So liver seems to be an important organ, and it goes back to almost 120, 130 years, the function of liver.
Great. Let's go a little bit to the biology of aging and to the hallmarks of aging. So you really nicely showed that taurine might influence all the hallmarks, or nearly all of the hallmarks of aging, including I think you showed methylation, but you also showed the senescence. And I would like to ask you, you cite that the mice had a lower number of senescent cells. Is that the cells do not accumulate, or is it that taurine even might have, while supplemented, a senolytic effect?
So we have not addressed this question precisely, to be honest, because we only looked at the end point here, where we looked at, through a variety of assays, accumulation of senescent cells after either vehicle supplementation or taurine supplementation. So during aging in mice, it was suppressing the number of senescent cells in aged mice after taurine supplementation. In zebrafish, for example, if you have telomerase deficiency, they had an increased number of senescent cells. And if you give taurine, senescent cells almost became to the baseline. And it actually rescued the accumulation of senescent cells and rescued zebrafish viability in this zebrafish telomerase deficient model. And not only that, we looked at DNA damage induced senescence and other upstream regulators of senescence. So it appears to me is that taurine somehow is making the vigor of the cells better and somehow acting downstream of these damaging processes that are happening upstream of senescence as a hallmark. So it could be, to me, it is more of a senolytic primarily and xenomorphic secondly. And that is what the data tell us. But we will investigate now in a preventive setting or in a curative setting what happens to the senescent cell accumulation after a different damaging processes.
Great. We know that senescent cells accumulation, or the number of senescent cells, is so much associated already with age-related diseases. So really looking forward to that. But staying a little bit at the hallmarks of aging, the epigenetic changes are also, during the aging process, are associated with lots of accumulations and incidences of age-related diseases in the end. Do you know how taurine is influencing methylation sites and GPT sites in the end? And do you know which epigenetic changes you see maybe in the animal models while being supplemented?
So one mechanism that we are although investigating, but I can tell you, is that the taurine deficiency, when you reverse it by supplementing taurine, it dramatically changes the ability of the cells to produce S-adenosylmethionine. And it is opening up DNA, or the chromatin is changing the landscape of chromatin by regulating the DNA and histone methylation. But in the liver, the DNA methylation pattern of aged animals supplemented with taurine was closer to the aged animals, not to the young animal. In the muscle, it reversed it. In the brain, it reversed it. But so it appears to me that the changes in the methylation pattern or the epigenetic changes overall, we classify them as, are not consistent or are not do not play the same role in different tissues. And I personally believe that epigenetic changes are still an evolving area, because we do not have a precise epigenetic clocks, for example, for a variety of tissues. In the blood, it is very well classified. And other tissues, it is still in a very nascent stage where we cannot say that the changes in methylation would tell you whether the epigenetic age was changed.
Yes, absolutely, fully agree with you that it's really organ specific in the end, the epigenetic clocks, and so dependent on the material you have. Let me ask your nearly final question before going to the audience. I already see lots of questions here. You showed that the mice experiment showed a 10% or 12% increase in life expectancy, whereas you see in C. elegans a much, much higher effect of 20 to 30%. Maybe it's interesting also for the audience who are not so much in the animal models. Why do we so often see a much greater effect in C. elegans compared to other model organisms such as mice and rats and even monkeys?
I think the answer to this question is perhaps more straightforward than we tend to think, actually. The reason behind that is that as the evolution occurred, the complexity of organisms have changed. The blood vessels are the one that are taking these different molecules to different organ systems, and different organ systems are utilizing different repertoire of these nutrient species. And blood passes through different organs before it reaches the end point. Whereas if you look at the worms, they eat taurine and it goes to different organs, primarily by diffusion, and other means as well. I'm not a worm biologist, I will not be very specific there. And so the cells have a better availability of the taurine in this case to metabolize. But in humans or in other species, in the higher mammalian species, this increase in complexity leads to less bioavailability of these different molecules, only if you look at precise dose translation. So the doses that we have used are consistent across different organisms. The doses that we have used are equivalent to 3 gram or 6 gram per day for a human being, which is under a safe limit of a human being according to European Food Safety Authority. And we went to monkeys and translated that dose based on those translation. We went to mice, we went to worms. So the doses are consistent. And I would think that if you change the doses a bit, you may have a different responses in different organisms, for taurine at least.
Yeah, so you do first a phase 1 study now to see what dose maybe is a good one for the PK and PD models, yes?
Yes, for the we are now starting to see what are the different requirements. But good news is that taurine has already been tried in clinical trial for specific diseases such as obesity, and there is a beneficial effect on obesity. There does not seem to be any red flags or any side effects that have been reported in humans. So we have a lot of positive news, and hopefully we would, once we have this dose response in the pilot study done in humans, we will have a clearer idea of what kind of doses to go in a larger trial.
Okay, great. Thank you so much. Over to Luang Guan, who is our research fellow at the Center for Health and Longevity. Lily, what do we have from the audience? I see lots of questions.
Yeah, thank you, Dr. Vijay, for your impressive presentation. We do receive some lots of questions from the audience. So some questions are similar, so I'll just combine them to ask. So let's start with questions from Iva and Vincenzo. So is it known why taurine decreases during aging? And as follow-up question, is it due to declining pancreatic function?
So some studies have been done in the past, and there are indication that liver production of taurine decreases. And that is primarily the taurine producing organ. So I would think that liver, along with some other organs which contribute, such as pancreas as well, would overall this biosynthetic capacity is declining, and that could be one primary reason by which taurine may be declining with age, because as I said, it is not known at the present time how are taurine levels in vegan versus non-vegan human subjects. And these investigations would take us some time to conclude.
Okay, next question. So reading your science paper, it almost seems like taurine is an mTOR inhibitor. And do you think this can account for most of the benefits seen? And also the other question is, taurine leads to weight loss in your study in both mice and the primates. Does this explain the many benefits seen?
Okay, so there are two questions. Let me go to the mTOR first. Okay, well, it was this is a very good question. We had that in mind for almost 5 years now. So taurine is the amino acid, although it has a sulfonyl group, which does not allow it to get incorporated into proteins. One of the initial inclination for us was that, okay, if taurine is an amino acid, all amino acids, many of them at least, bind to mTOR complex and affect its function. The first thing we did was to look at mTOR, and like the question is here. So taurine did bind to mTOR complexes structurally in a bioinformatic, in a molecular modeling experiments we did. But when we did, no matter what kind of conditions we did in the cells, fed, fasted, does not matter. You put taurine supplement, taurine on top of the cells, it does not affect the mTOR activity directly. This data is not published because it's all negative data. We are trying to publish it somewhere. But taurine on the cells, when you look at it, it does not affect the translocation of mTOR across different cell populations we tested in vitro. So I do not think, based on the experimental evidence I have, that taurine affects the mTOR activity directly. It is acting, in my view, on a downstream process like autophagy, protein homeostasis, but not on mTOR.
Okay, how about the weight loss?
Weight loss, we did see some weight loss. Obesity was reduced, for example. There was a reduction in body weight. But weight loss happened after the beneficial effect of taurine was already present. For example, if you look at males did not lose weight up to 4 months. And we had health benefits of taurine supplementation in aged males by 4 months. And we investigated, but they had less adiposity. Body weight was not changed, but adiposity was less. And we investigated the reason why they had less adiposity. And it was due to a very high increase in energy expenditure after taurine supplementation. So we think that the mild body weight, 7 to 8% reduction in the females that we have seen, or little bit less in males that we have seen at a later point in taurine supplementation studies, is due to changes in the entire body composition. And there is already health benefits apparent before body weight loss is seen.
Thank you for your answer. And next question is regarding the dosage. So the doses you use are very high compared to clinical used doses, and even adjusted for body surface area, yet they barely normalize taurine levels in old mice. This is very interesting. And is there an issue with absorption? And the other question is, might there be any interaction with other drugs by taurine supplements? Which dose could be delivered with safety when needed?
So I think I briefly touched upon this issue. In humans, up to 6 grams of taurine has been considered under safe limit in official documents by European Food Safety Authority. And the doses that we have used in mice are 3 gram equivalent or 6 gram equivalent of taurine. That is 500 milligram per kg body weight or 1000 milligram per kg body weight per day of taurine in mice. So the doses are under the safe limits of taurine supplementation. And one reason why taurine is seems to be safe in humans, it is because any excess that we take, which is not taken up by cells, is quickly excreted in the urine through kidneys. And that keeps the homeostatic level of taurine in the blood to a reasonably constant level, because cells have either taken up taurine or whatever was excess was taken out by kidneys in the urine.
So are there interaction with other drugs?
We don't know the answer to that question because no investigation has been done in humans to address this question. And as I said, before we embark on a large clinical trial, we are looking at identifying the healthy reference reagent of taurine in different ethnicities and different population, different metabolic processes regulating taurine biosynthesis or its downstream degradation. And these are the questions which I think will get clear up in next year or so, in my view, before we have the clinical trial on the go.
Okay. The next question, so the audience is interested in the exercise results. So does exercise increase endogenous taurine production in the body, and would that bring health benefits compared to the levels needed via supplementation too? And the other audience also asked, what are the mechanisms increasing blood taurine concentrations after exercise?
The straight answer is I don't know. We do not know whether exercise is increasing taurine levels by increasing biosynthetic capacity, or by increasing the release of taurine that is stored in tissues. I would tend to think it is the second, because just after an hour of exercise, you have a major increase of taurine levels in the blood. And changing taurine biosynthesis in such a short span of time is it's possible, but I think it will be more likely it is released from the tissues that have stored it, because taurine is mostly present as a deuterion or a free molecule within the cellular cytosol. So I would tend to think that it is the release from the organs that contributes to increase in taurine after exercise.
Next question is from Colin. So as in your graphic shown, taurine supplementation leads to hydrogen sulfide production, and given that dietary restriction also increases hydrogen sulfide, and which is required for longevity, thus what is the contribution of H2S to mediate these health benefits and longevity upon taurine supplementation?
Well, this is an important question. I don't know the answer to it. Yes, hydrogen sulfide production is increased in some studies that I have seen by the gut microbiome upon oral taurine supplementation. But how it is going to contribute to the increase in the health of an organism, because H2S is very short-lived and it acts in a very neighboring cells more than anything. So the contribution of this has to be investigated properly, and perhaps H2S production by mammalian cell need to be investigated, how it is affecting the mammalian production of H2S. Can it be metabolized? And these are the questions that need to be addressed.
Next question. As we know, there are hallmarks of aging, at least 3 categories: primary, antagonistic, and integrative. which one of this category mainly affected by taurine?
So as I said, the hallmarks are a vague classification of looking at aging, in my personal opinion, because these are just cellular features which are changing with age. So everything changes with age. So you can make 100 hallmarks of aging, for example, DNA replication or RNA replication or RNA synthesis or RNA degradation. And all of these change with age. So, and all these hallmarks, so-called hallmark, interact with each other. There is no hallmark I know of that works in isolation. If you have DNA damage, oxidative DNA damage, for example, it leads to increased accumulation of senescent cells. If you have telomerase deficiency, it leads to increased senescence, increased accumulation of senescent cells, and causes DNA damage beforehand. So the hallmarks are not working in isolation. Mitochondrial dysfunction or oxidative stress, all these are interdependent processes. We cannot look at one hallmark, antagonistic or agonistic, in my personal view, to define effect of an intervention. We have to look at cellular processes. We classified them into 9 hallmarks because that is what is available in literature and is well received in terms of how to look at the aging process. But taurine or any other intervention is affecting a lot of processes, including rapamycin, for example. It is not only acting on mTOR, it is acting on a variety of other processes as well. mTOR is just more popular.
Yes. Okay, next question from Jeremy. Understanding normal taurine levels is complex, as taurine is used in many consumer-facing products already, like Red Bull, supplemented waters, energy drinks, etc. And the evidence appears to be association. What work is being done to understand the causation rather than looking at the case control studies? And that he feels this is going to confuse matters.
No, we are absolutely, I agree with it. We don't have a causation, and that is why we are doing starting a clinical trial, putting it together to test in a randomized placebo-controlled clinical trial in humans to see the health benefits of taurine. Is it a driver of aging in humans too? I don't know the answer to that. And till then, I don't recommend any taurine supplementation.
Okay. As you mentioned, the clinical trials, so our audience would like you to maybe elaborate more on the design of the trial being planned.
Yes, so we are what we are going to be looking at the present time is the health span. After 1 year or 2 year of taurine supplementation, we are going to looking at a variety of health markers of muscle function, bone function, liver function, brain functions, and kidney functions. And that is what we are trying to come up with, the series of different endpoints which can be realistically measured in humans, and that has to be harmonized across different population we are envisioning. We need to look at these standardized markers of health across different populations. Same markers will tell us how it is changing. Is it different ethnic groups respond differently? And whether male and females will respond differently? I think these questions will be very clear after post-hoc analysis of those data.
Okay. Maybe I'll just ask one more question. So taurine is also an antioxidant. Did you investigate this aspect with aging benefits in the model organisms?
Yes, taurine is an antioxidant. You're right. It, at least in the immune cells, it is a major antioxidant. It neutralizes the hypochlorite that is being produced by the immune cells, and making taurochloramine. And also in these cells, it is somehow scavenging the oxidative species, reactive oxidative species that is being produced. I don't know the mechanism of it at the present time, but at least in the immune cell, we know that it binds to the hypochlorite and making taurochloramine.
Thank you so much, Vijay. What a wonderful talk. And I liked your comment you just made that please the audience do not buy now the supplements in Taurine and just supplement yourself. Is that right? That's not what you suggest.
Absolutely, I agree. And it is true for any other anti-aging intervention. We cannot take over-the-counter medicines and supplement ourselves because the clinical trial for most of the aging interventions are on the go at the moment, in my view. And next 3 to 4 years, we will have all these molecules in the anti-aging basket, which people can choose from, because we are in the era of precision medicine. We are no longer one shoe fits all. Let us make all the horses run and see which one runs better or which one works better for a particular organ function before clinicians can prescribe these anti-aging interventions. And that is what needs to be done.
Thank you so much. Really a virtual applause. So I think this talk and all the others, as always, triggered lots of questions. Please leave us your comments and suggestions. We're really happy to have them. Use the panelists and all attendees option. There are a couple of announcements. First of all, there is the Save the Dates. Yes, we are hosting the next conference, Unlocking Healthy Longevity, just focusing on supplements. The first only the conference just focusing on supplements as a geroprotective intervention. The second announcement I have, the Academy for Healthy Longevity offers before that conference at the 29th of February to the 1st of March next year, a mid-career class before that conference. So watch the space. The third announcement I have, I get lots of emails and questions. How can I watch all these nice presentations and webinars back? Yes, you can. We have a YouTube channel. So look at the chat box and you will find the link. It's my honor that at the 3rd of August, I will host Professor Vadim Gletchev. He is professor of medicine at the Harvard Medical School, and he will talk about quantifying aging, rejuvenation, and longevity. As always, there is a final video, and this time I would say a little bit of laughter always helps. Take care.
All men get to this age. Now, I don't know if women ever get to this age. Let me see. Of course, you're not there. You're not there. I can't tell if you're there. You're there. And this guy, you've been there for a while. Now, what age am I talking about? I'm at the age I'm starting to make noises when I don't want to make noises. If I do any kind of work around the house, I make 2 noises. One noise when I do the work, and then another noise is about a second later. And it sounds like I'm thinking about the work that I just did. Now, I'll show you, and you'll get it. Watch this. Okay, you ready for this? Here we go. Everyone get it? Are we all on board? That's the whole joke right there. Everything 2 noises. I move a lamp in my house, a little floor lamp. I make noises doing almost nothing. Sitting down. I come home at the end of the day, I sit in my chair at the end of the day. Woo! Woo! Have you ever sat down, made a noise, and had people concerned for your well-being after you make the noise? Woo! Are you okay? Are you all right? I'm fine. Thank you very much. I got my butt all the way back. Thank you.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.